In this video tutorial, I am going to discuss about regulation of glycogenolysis and glycogenesis. We know that glycogenolysis is the pathway in which glycogen undergoes breakdown to first give rise to glucose 1-phosphate, which then gets converted to glucose 6-phosphate, and glucose 6-phosphate gets converted in the liver to glucose. Whereas if it is in the skeletal muscle, then this glucose 6-phosphate cannot get converted to glucose, so this undergoes glycolysis and it will provide energy to the skeletal muscle. This pathway of glycogenolysis, its main regulatory as well as rate limiting step is catalyzed by the glycogen phosphorylase. Whereas the reversal of this pathway, that means polymerization of glucose or glucose 6-phosphate to synthesis of glycogen, this pathway it is called as a glycogenesis. And here the regulatory as well as rate limiting step is glycogen synthase. As you can see that both of this pathway, glycogenolysis and glycogenesis, both are opposite pathway. So we have to discuss regulation of this both pathway together, then only we can have an idea about the complete picture of regulation of glycogen metabolism. Now before discussing about the fine details of this regulation of glycogen metabolism, let's first look at the bigger picture. Here we know that this both process, glycogenolysis and glycogenesis, both process occurs in all the tissue. However, maximally it occurs in the liver and skeletal muscle. In the liver, the main role of glycogen metabolism is to maintain blood glucose level. Whereas in the skeletal muscle, its main role is for the provision of energy for the contraction of skeletal muscle. Let's first look at the liver. In the liver, what happens whenever we are in the fed state, that means we had just taken food. So because of the digestion and absorption of carbohydrate, our blood glucose increases. This increased blood glucose, it leads to certain regulatory events. It will ultimately lead to increased glycogenesis. And we know that in the glycogenesis, glucose will be utilized for the synthesis of glycogen. So there will be decrease in the blood glucose. Opposite will occur whenever we are in the fasting state. Whenever we are in the fasting state, there will be decreased blood glucose. This decreased blood glucose leads to certain regulatory event and it will lead to increased glycogenolysis. And because of the glycogenolysis, glycogen will be broken down to glucose. So that will lead to increased blood glucose. So as we can see over here, that by these regulatory events, increase or decrease blood glucose is leading to decrease or increase blood glucose. So that's how blood glucose is maintained. What happens in the skeletal muscle? Now whenever skeletal muscle, they are doing exercise or undergoing contraction, so exercising skeletal muscle, they need energy. So exercise will lead to certain regulatory event which stimulates this breakdown of glycogen. So there will be increased glycogenolysis. So by this glycogenolysis, there will be more production of glucose 6-phosphate and so it will undergo glycolysis to provide energy to the skeletal muscle. The opposite will occur whenever skeletal muscles are at the rest. So rest will lead to certain regulatory event and it will lead to increased glycogenesis. So this is the bigger picture. Now in the finer detail, we will discuss that how these regulatory events are taking place. So for discussion of this finer detail, this regulation of glycogen metabolism, it occurs by two different ways. One is allosteric regulation and second one is the covalent modification of enzymes. Now covalent modification of enzymes, it can be carried out by two different molecules. It may be by hormones or it may be by calcium ion. In case of hormones, we have to discuss three very important hormones. One is glucagon, second one is epinephrine and third one is insulin. Here covalent modification is in the form of phosphorylation or dephosphorylation. And these enzymes which are affected by this covalent modification are glycogen phosphorylase and glycogen synthase. So let's start our discussion of this regulation by first discussing this allosteric regulation. In case of allosteric regulation, we have to consider four important molecules. One is glucose 6-phosphate, second one is adenosine triphosphate, third one is glucose, and fourth one is adenosine monophosphate. These four molecules are very important allosteric regulator of glycogen metabolism. But before that, understand glucose 6-phosphate and adenosine triphosphate both occurs in both the tissue, liver as well as skeletal muscle. Whereas glucose, it occurs only in the liver, whereas adenosine monophosphate, it is allosteric regulator only in the muscle. So with this information, let's see how these four molecules are affecting these two different enzymes. Glucose 6-phosphate, ATP and glucose. All these three molecules, they are important allosteric inhibitor of glycogen phosphorylase. Whereas adenosine monophosphate is important allosteric activator of glycogen phosphorylase. Whereas for the glycogen synthase, only glucose 6-phosphate acts as an allosteric modulator of this enzyme, and this modulation is in the positive direction. That means glucose 6-phosphate is the allosteric activator of glycogen synthase. Now many times students find it difficult to remember which compound is found in both the tissue, which compound is found in only liver, and which compound is found only in the muscle. So see over here that glucose contains L, so it is found in liver. Whereas adenosine monophosphate, it contains M, so it is found in muscle. Whereas remaining molecules, they will be found in all the tissue, liver as well as skeletal muscle. Now let us try to link this finer detail with this bigger picture. See what happens. In fed state, there is increased blood glucose. Now this increased blood glucose, it will lead to increased glucose in the liver and increase glucose 6-phosphate. Inside the liver, this both glucose and glucose 6-phosphate, they inhibit glycogenolysis. Whereas this glucose 6-phosphate, it activates glycogen synthase. So what will happen? There will be glycogenesis. So here we can see there is increased glycogenesis, which will help to bring down blood glucose. The opposite will occur in the fasting state. Whenever there is decreased blood glucose, at that time this allosteric inhibitor action will be gone away, and this glycogen synthase, its stimulation will go away. So the net direction will be towards the glycogen breakdown. So it will lead to increased glycogenolysis, and this will lead to increase in the blood glucose. Let's look at the muscle. In the skeletal muscle, when they undergo exercise, so during exercise, energy is required. So what this skeletal muscle will do? They will break down ATP to ADP and AMP. And energy will be released that will be utilized for the skeletal muscle. Now because of this, ATP's concentration will decrease and AMP's concentration will increase. See, this AMP in the muscle, what it is doing? It is activating glycogen phosphorylase. That means there is an increased glycogenolysis. So by this increasing glycogenolysis, glucose 6-phosphate will be increased, and so there will be increased glycolysis, which will provide ATPs, right? And these ATPs will get replenished. And opposite will occur whenever we are in the resting state, right? So now this completes allosteric regulation. So now let's look at the covalent modification of enzyme. First we'll look at the action by hormones. So we have three important hormones. The first one is glucagon. Second one is epinephrine. And third one is insulin. Now to understand this, suppose this is the cell membrane. On the cell membrane of liver cell, there are glycogen receptor. So glycogen receptor, they are present only on the liver cell, whereas the receptor for the epinephrine, they are called as a beta receptor. They are present in liver and skeletal muscle. Whereas the receptor of insulin, they are present in all the tissue. Remember, hormone cannot enter inside the cell, so they binds with the receptor. So when glucagon binds with its glucagon receptor on the surface of hepatocyte and epinephrine binds with its receptor beta receptor on the liver and skeletal muscle. This ligand receptor complex leads to activation of one membrane bound enzyme which is called as a adenylyl cyclase. So this adenylyl cyclase will get activated. And what is the function of adenylyl cyclase? It will convert ATP to cyclic MP. This cyclic MP is very important molecule that we will see in a very short time. But before that, cyclic MP can be broken down to 5' MP by one more enzyme which is present in the cell, that is phosphodiesterase. This phosphodiesterase is often abbreviated as a PDE. Now, why this cyclic MP is very important? The reason is that cyclic MP, it stimulates cyclic MP dependent protein kinase A, and it is often abbreviated as a pKa. Now, this enzyme is very important as far as glycogen metabolism is concerned. What this cyclic MP dependent protein kinase A will do? Cyclic MP dependent protein kinase, it causes phosphorylation of glycogen synthase. So this is the glycogen synthase in its dephosphorylated state. It gets phosphorylated by the action of cyclic MP dependent protein kinase to glycogen synthase in its phosphorylated form. Meanwhile, it also carries out phosphorylation of one more enzyme, that is phosphorylase kinase. So phosphorylase kinase, from its dephosphorylated state, it gets converted to phosphorylase kinase in its phosphorylated form. This phosphorylase kinase, what it will do? It will carry out phosphorylation of one more enzyme, and that is our glycogen phosphorylase. So this is glycogen phosphorylase in its dephosphorylated form, and this is glycogen phosphorylase in its phosphorylated form. So this phosphorylase kinase, it will catalyze the conversion of phosphorylation of glycogen phosphorylase. Here in this case, one thing which is very important to note down is that glycogen synthase is inactive in its phosphorylated form, whereas its dephosphorylated form is active. Whereas it is opposite in these two enzymes. Phosphorylase kinase is inactive in its dephosphorylated form, whereas it is active in its phosphorylated form. And similarly, glycogen phosphorylase, it is inactive in dephosphorylated and active in the phosphorylated form. And there is one convention as far as glycogen metabolism is concerned, is that whatever the active form is there, they had given a tag of A. So here this form is active, so we call it as a A form. Here this form, this phosphorylated form is active, so we can give a tag of A. Here in case of glycogen synthase, this form is active, dephosphorylated form is active, so we give a tag of A over here. So here A is for the active. Whereas the other inactive form, it is given a tag of B. So here this inactive form will be called as a B, this inactive form is called as a B. Now here many times students find it's very difficult to memorize which form is active, phosphorylated or dephosphorylated for particular enzyme. So for that, I had one very short and simple trick. So as far as glycogen metabolism is concerned, whenever there is a phospho in the name of any enzyme, they will be active under phosphorylated state. See here, in case of phosphorylase kinase, there is a phospho in the name, so it is active in the phosphorylated state. Glycogen phosphorylase, it has also a phospho in the name, so it is active in the phosphorylated state. Whereas glycogen synthase, there is no phospho in the name, so it is active in the dephosphorylated state. So by this mechanism, you can easily remember which form will be active. And remember, this trick is only for the glycogen metabolism. This is not general trick. So now let's try to understand that what is the final effect of this glucagon and epinephrine. See, whenever glucagon and epinephrine, they are present, they will lead to increased cyclic MP, they will lead to activation of this protein kinase A, and by that, glycogen synthesis is getting deactivated. That means there will be decreased glycogenesis. And glycogen phosphorylase is becoming active. So that means there will be increased glycogenolysis. Now all these three enzymes, they can get dephosphorylated by one common enzyme. So all three can get dephosphorylated by one common enzyme, that is phosphoprotein phosphatase 1. This enzyme causes dephosphorylation of this glycogen synthase. The same enzyme carries dephosphorylation of phosphorylase kinase, and this same enzyme causes dephosphorylation of glycogen phosphorylase. Now, this cyclic MP dependent protein kinase has also effect on this phosphoprotein phosphatase. What is the effect? This cyclic MP dependent protein kinase, it phosphorylates one more protein, that is inhibitor 1. This inhibitor 1 protein gets phosphorylated by this action of this enzyme. So inhibitor 1 in its phosphorylated form. And what this inhibitor 1 protein will do? This protein will inhibit this phosphoprotein phosphatase 1. So by this, what is happening? So on the one side, it is causing phosphorylation of all these three molecules. And on other side, it is inhibiting dephosphorylation of all these three. So that means the ultimate effect is that all these three enzymes will be kept permanently phosphorylated by this double side action, so that the glycogenesis will be almost suppressed and glycogenolysis will be very much stimulated. Now let's try to link this minute, this fine detail with this bigger picture. See what happens whenever we are in the fasting state, there will be decreased blood glucose. So decreased blood glucose, it stimulates release of glucagon from the pancreas. So this glucagon comes to the liver and binds with its glucagon receptor. Then it will lead to all these regulatory events, which ultimately lead to decreased glycogenesis and increased glycogenolysis. So that is written over here that there is increased glycogenolysis, which leads to increased blood glucose level. So here liver is fulfilling its role of maintaining blood glucose. Now let us look at here skeletal muscle. What will happen in the skeletal muscle? In the skeletal muscle, whenever there is a fight or flight response, that means whenever you are under panic situation, suppose you are roaming in the forest and suddenly lion comes in front of you. So at that time, either you fight with the lion or run away from the lion. That is fight or flight. So under panic situation, in any condition, muscle contraction will be required. So during this condition, epinephrine will be released from the adrenal gland. This epinephrine comes to the skeletal muscle and binds with its beta receptor. And that will lead to sequence of event, and it will ultimately lead to decreased glycogenesis and increased glycogenolysis. So by increasing glycogenolysis, what will happen? Glycogen will be broken down to glucose 6-phosphate, and this glucose 6-phosphate undergoes glycolysis in the skeletal muscle, which will provide energy for this skeletal muscle contraction during fight or flight response. So here this both minute details we understand in this from this bigger picture. Now, when we are discussing so much about glucose and glycogen, how can we forget insulin? Insulin binds with its insulin receptor, and it will lead to some sequence of event which ultimately stimulates phosphodiesterase enzyme. So by stimulating phosphodiesterase, what will happen? Cyclic MP will be broken down to 5' adenosine monophosphate. So there will be decrease in the cyclic MP. So there will be all the effect which are carried out by glycogen and epinephrine, they will get reverse. Insulin also acts on this phosphoprotein phosphatase. It will lead to sequence of regulatory reaction, and it will stimulate phosphoprotein phosphatase. So by this, what will happen? This dephosphorylation will be activated. So glycogen synthase will be activated, and this will lead to increased glycogenesis. Whereas glycogen phosphorylase will be inhibited. So this will lead to decreased glycogenolysis. So by this both process, it will lead to increased glycogen synthesis. So insulin, it is secreted in the response to increased blood glucose. This increased blood glucose lead to release of insulin from the pancreas cell. It will come into all the cell, and it will stimulate phosphodiesterase, And so cyclic MP will get decreased, and it also causes dephosphorylation of all these 3 enzymes. And by that, there will be increased glycogenesis in which glucose will be utilized. So it will lead to decreased blood glucose. So here insulin is also regulating blood glucose. Now last but not the least that we have to discuss is the regulation by the calcium. Now this occurs predominantly in the skeletal muscle. See, whenever skeletal muscle undergo contraction, at that time from the sarcoplasmic reticulum of the skeletal muscle cell, they release calcium ions. This calcium binds with one protein which is present in the cytoplasm of the skeletal muscle, that is calmodulin. In this calmodulin, this is cal for calcium, module for modulating, and in is the last 2 alphabets of the protein. So calcium modulating protein. This calcium binds with this calmodulin and forms the calcium calmodulin complex. It is also abbreviated as a CAM complex. This CAM complex directly activates this phosphorylase kinase, and we know that this phosphorylase kinase, it causes phosphorylation of glycogen phosphorylase. So there will be increased glycogenolysis. So here also you can see or appreciate the fact that skeletal muscle when undergo contraction, there is increased glycogenolysis. So this will lead to increased glucose 6-phosphate, which will increase glycolysis, and so ATP production will be there, which will be utilized for the skeletal muscle for contraction. So this completes our all discussion about the regulation of glycogen metabolism. In the next video tutorial, I will discuss about glycogen storage diseases. If you have any query or confusion, please write it down in the comment section below. Thank you.